And now she's also involved in developing tools to make pangenome accessible to all the people working in genomics and genetics. She contributed to several very important projects, like the 1000 Genome Project, like the Human Pangenome Reference Consortium, so helping to build resources available to all people studying genetics and genomics. She's also very, let's say, committed to train students. And I'm one of the product of this, so she's very good, I can say, at the end. Also, she founded and organized the I think 9 editions of the EMBO population genetic course. She's also she organized a lot of, for 3 years now, I think, the pangenome course here in Memphis. So what else to say? I can just, today she's going to present something about integrating genetic ancestry, environmental exposure, and health equity. And here.
Did you mention the bats?
No, I wanted to say that I said she's interested in human genetics, but not anymore. Every day she comes to the lab and she takes a new species. She forces us to study new species, but just I didn't want to say bad things about you. So I'm leaving and the stage is yours.
Well, Sylvia, thank you, Sylvia. I think Sylvia wanted to say that I'm so committed to training that I forced them to work on what I call training projects, like the bats and things like that. But yeah, anyway. So yes, so so it's a real pleasure to talk here today. I'm kind of like really thankful for this opportunity to present the work that we've done in the past. I think it's 2.5 years, and it's really nice to see it all while I was making the presentations, just to see everything coming up together. And so my first my first thoughts goes to people in my lab who make this possible. I have to say they've been exceptional. And so all you can see here is actually their work. And for some reason, they asked me to present it, but it's actually work from, and you're gonna see credits to everyone. Okay, so just to put this into context, we are in Tennessee, and everybody knows that. But what you probably don't know is that Tennessee has been ranked 48 of 51 in terms of like state health system performance, which is not a good news at all. This is also 2023, but I don't think things has changed much. And also, one thing that you probably are aware of is that so Tennessee is one of the least diverse states.
Yeah.
It's one of the least diverse states where 71% of the population is mostly self-reported white. However, Memphis, which is home of the famous Mem Pops, which is happen to be also my favorite one of my favorite things in Memphis, it's different. And we do have a prevalence. It's kind of like something that really outstanding from the rest of the Tennessee. And we have a prevalence of Black or African-American of about 51%. So why I'm starting with this, because I want to put the work that we do into the social context where we operate it. So within this context, the Biorepository for Integrative Genomics is an initiative that started way before I was like affiliated with GGI in 2015. And so far has enrolled about 45,000 individuals. We are, very close to hit the milestone of 50,000 enrolled individuals, for which we have electronic health records. We collect leftover blood, so we have collected only 20,000 biospecimen because there's not always leftover blood, and for several reasons. And then we sequence 13,000 of those like collected samples. And we, at the moment, we're collecting mostly at Lebanon Children's Hospital and Regional One Health, which is like a regular adult hospital in Memphis. But we also receive samples from East Tennessee University, which is like located at the upper corner of Tennessee. And we also have some other donation from other collections. This is great, but so in 2004 2004 I started my commitment also as director of the biorepository. Since then, I've been recording how many samples we collect, and I'm really proud to say that we collect about 100 samples per week, and that translates in about 5,000 samples per year, which is a lot. And this morning I was talking with a student, and they were talking about the study design. The idea is to get as much samples as possible, as fast as possible. So that's the way we're set. And here is Donna, which is the lab technician. She's mostly in charge of the lab and the sample collection. And Ernestine occasionally would, she couldn't come today, but Ernestine occasionally would help. So our cohort is mostly pediatric, but we are moving toward, we kind of exhausted the pool of pediatrics kids that we can recruit. And we are now recruiting more and more adults at Regional One Health. What is important is that BIG is not an isolated initiative. We're part of a federated initiative, which is called Genomics Information Common. And you see here on the map the different institutions and the different places. The idea is to have a federated way of collecting data so that whatever funds we have in one of the participating center can be replicated into other hospitals. And this is mostly a network of pediatric institutions. I'm really proud to say that we also so the picture didn't show up, but we are part of Together for Change. We signed the contract, we closed the contract. We're going to be part of this initiative, which is aimed to create a database of people of African ancestry to address underrepresentation in genetics. And the key partners, the Meharry Medical College, which is leading the initiative. And the initiative is founded by this four pharmaceutical company. And I'm worried we don't see images here, I hope. Anyway, so which kind of data we have? So the primary source of data is genetic data, social determinant of health, clinical data, and most important, zip codes. And you'll see this why this is very important. And then from this, especially from the genetic data with my group, we worked on infer some other type of metadata like the ancestry. And I'm going to talk about that. And this has enabled, oh yeah, before you ask, yes, the data is available. We are really willing to share the data. We encourage people to use and reuse BIG data. And, and there's a simple link and there's a simple way to ask for the data. But what I want to emphasize here is that within this past 2 years, we have been able to, thanks to this metadata, to sharing this metadata that we produce, we've been able to enable full collaboration, one of which has already been published. So I'm really proud to say that whatever BIG is building can really enable a network of collaboration. Okay. So this was advertised for BIG. Now let's talk about science. So the first question when I started working on BIG was like, everybody was saying, oh, we have a really diverse cohort. And I was like, how much diverse? And it was like, oh, people self-report. And I was like, they self-report, but actually how much is this diverse? So that's the first thing we did. And that translates into determining ancestry, to do something that is called ancestral deconvolution. Sorry, let me find my mouse. Why I don't see? Interesting. Do you see it? Okay. So if you're not familiar with the concept, the idea is to guess for every individual's, for every chunk of the genome, where the genome is likely to come from. And so imagine, you have parental genomes, which are of two different populations. And then the F1, of course, is going to have like one set of chromosomes from one population, the other from the other. But then, like, by the time passes and recombination takes place, then the genome of current mixed population is just a mosaic of different chunks from different populations. And that's because recombination just reshaped the way our genome is made. So the challenge is to get what we have is people here at FN, and then look at the genome and understand from which ancestry they are likely to derive. And we did that. Actually, they did that. And this is the results. What you see here is a plot with 13,000 vertical lines, which you cannot distinguish because they're very tight. And the colors represent the likely origin. And what you see here that there's a big chunk of orange people, which has been assigned to be very similar to European. Their genome is entirely likely to be of European origin. And then there's a bunch of people here who is actually almost entirely blue. That means, they're very likely to be African origin. But also there's a bunch of people who is admixed. That means their genome, it's half coming from European ancestry and half coming from African ancestry. And that's really exciting because that's, the first conclusion that we could get from that is that 50% of the BIG cohort is of non-European origin, which is like a good news because qualify us for participating in this initiative about underrepresented genomics and underrepresentation of certain population in genomics. We have different type of admixture between European and African, admixture between European and Native Americans, and then also multi-way admixture, which is like a little bit of everything. But what is striking it? So that's, that was the first thing that we were able to determine. But then because we had the zip code and we could map the individuals on a geographical map, Franco was curious to say, well, let's see how this ancestry are distributed on the geographical map. And the results was like, wow, if you've been in Memphis for a few months, you figure it out yourself. But if you're not, this is, incredible. So that's the prevalence of European, of individuals of African ancestry. And that's the prevalence of individuals of European ancestry. And they look like a puzzle. They're just perfectly complementary, which means that the communities of individuals are highly segregated. We also wanted to know, there's Ernestine joined the lab a year and a half ago, and she was, interested in working on BIG. And we said, what about the mitochondrial DNA? Because as you know, the mitochondrial DNA, it tells a different story. It's the story of the maternal lineages. And we said, let's determine the geographical origin of the mitochondrial DNA. And let's compare with the ancestry that we inferred. And what you see here is that, for instance, the individuals that we inferred to be Europeans, their mitochondrial DNA is mostly of Western Eurasian origin, so which makes sense. And the same for individuals that we infer to be of African origin, the mitochondrial DNA is mostly of Sub-Saharan African origin. But we want to highlight two cases. This is the admixed populations. So individuals that we classify as admixed between European and Africans, they actually, someone want to enter, Megan? Someone. So we would expect this to be 50% teals and 50% purple, meaning that their mitochondrial DNA would come 50% from African origin, 50% from European, but actually that's not the case, not at all. And this difference, this discrepancy from 50-50 is actually significant. And that means, and this is the same for the European Native American admixed individuals. And that means that there have been sex bias admixture. That means that in the admixture event, the contribution was not equal. There was a different number of female and male for every ancestry. And this is something also that everybody knows. And, just like it's been demonstrated also for other populations. So we went a step further on that. And we wanted to kind of reconstruct the demographic history of this sex bias admixture. And this is the first time I'm presenting this slide, and it's going to be a pain, but it's going to be a cartoon that, make it easier at the end. So bear with me. So this is the affected population sites of the Sub-Saharan mitochondrial DNA lineages. And this is the European mitochondrial DNA, and everything makes sense. So if you're not familiar with population genetic, affected population sites is a measure of genetic diversity. The larger it means, the more diverse is the pool of sequence that you have. And so that means that the African mitochondria, in the admixed individuals, the African component is very rich. There's a lot of genetic variability in the mitochondrial DNA. However, the mitochondrial DNA allows fast reconstruction of affected population size through time in an interval of years, which goes very deep. This is like today, and this is like 100,000 years ago. And you see the population, was small, and then, we all know that human population expanded, and it all makes sense. But we can't say much about very recent time. And if we all know that if admixture happened, it was during the forced migration after, the discovery, after America was colonized by Europeans. And so we used genomic DNA to investigate this much recent window. And so with genomic DNA, we can reconstruct, affected population sites up to 2,000 years ago. And that's a much, it's a magnification of current time. That includes the window of time in which forced migration happened. And what we see here, if you follow the blue line, the solid blue line and the solid orange line, this is the affected population sites of the African is the blue and European is the orange component of the admixed individuals. And the picture that we got is that there were few European, because this this is smaller, few Europeans admixing with a lot of African, sorry, it's the opposite. I'm going to show the cartoon because it's easier. It's really the first time we're showing this result, so I'm really proud of it. So this is, in short, what happened. In the admixture event, what we can say, there were a lot of European males admixing with few African females. And that makes complete, that really makes sense with what the history that we know. Okay, let's move forward. This was cool. Someone else want to be admitted? Okay, so this is about the demography and about the diversity. But did we find any interesting genetic variants? Because we have sequenced like 13,000 individuals with peculiar demographic history, which are so different from what we usually have been sequenced so far. So did we find anything interesting? And the answer is yes. And I'm going to show this. So this is just like the number of variable sites per genome. And this is exactly as we expected. So the individuals with African ancestry actually have the largest number of genetic variants per genome. And if you wonder about the scale, Eric, this is exome, not whole genome. So that's why we have tacits. So this is actually something that we measure in the 1,000 genome. And I have like, and that's, it's just like as expected. However, we did discover novel variants, and novel variants were under 11% of the variants that we discovered. They were mostly, shared, but there's a portion of these variants which were private. And guess what? They are found mostly, so the ancestry with least novel variants is Europeans, of course, because, we sequence a lot of them. Whereas the ancestry with least novel variants, in which we found a lot of novel variants, are actually the admixed, the Native American, and the Africans. And also, so we annotated the novel variants. Well, Sylvia annotated the novel variants to understand if they are actually able to have an impact on the gene function. And you have 4 categories here, and you cannot really appreciate the difference in the impact in between known and novel variants in the eye. But we evaluated that is actually novel variants have a high impact twice compared to the novel variants. So that means, I said it really bad, but what I wanted to say is that like novel variants are twice as likely to have a high functional impact. So we are actually discovering genetic variation that haven't been studied before, and is likely to have impact, as far as we know, on gene products. What about rare novel variants? Something really interesting came up. So this is the number of rare novel variants per gigabase in the genome. And what is standing out, and this is known also in similar, like other studies also found the same, is that what we did here was to measure the rare novel variants in the Native American portion of the genome of the admixed European Native American individual. So the purple is that value in the Native American part of the genome, and the orange is that value in the European part of the genome. And we do have a lot of novel rare variants in part of the genome that haven't been studied before. That's the main message. So first of all, I'm glad we can really dissect the genome of one individual into pieces and tell something which is ancestry specific. And what we found is exactly what we expected. You know, there's a lot of rare novel variant in population that haven't been studied before. What about rare deleterious variants? So let's add another level. So rare variants which are also deleterious. So we compare the rare deleterious variants across ancestry. And to do that, we normalize by synonymous because I show you that some ancestry just has a lot more variants. And the synonymous variants, if you're not familiar with that, it's variants like with neutral impact, like, you know, which are supposed to be harmless. And again, what we see, and it's already been described in our similar population, is that the admixed population, they have the highest large number of the deleterious variants. And in particular, for the European Native American, we counted the number of rare deleterious variants per gigabase, and we counted them in the American portion of the genome and the European portion of the genome. And we find a higher prevalence of rare deleterious variants in the Native American portion of the genome. And then we said, should we publish this? And people's going to say that, you know, Native American, they have a lot. You know, this is a measure of genetic burden. This is a measure of how likely your genome is like broken and there's harmful variation. And so what we're saying here is that the Native American portion of the genome of the admixed individuals has a higher burden. And as a population geneticist, I say we explain this because of demography. And also it has been observed by other like study on admixed populations. Yes.
Not to let this go, because I think this is a very important point. Deleterious is an imputation from looking at sequence.
Yes.
Not a phenotype.
No.
So it should be deleterious with double, triple.
What's yes. Yes, thank you. Thank you for clarifying. Yes, and that's, you know, something we debated a lot before publishing a function, potentially functional effect on the gene. So yes, this is a long, and that's also why, you know, we wanted to dig further into this, but then we decided, oh, it's too much, because like, how do you define? That's exactly what you're saying, Rob. We don't have.
Hemisphere. They have a stop codon in a gene that all other birds have. Do hummingbirds have a deleterious stop?
No, they're probably fine. Yeah, no, it's just.
No, that it's actually essential for hummingbirds to beat their wings at 20 hertz.
Right.
You can't do it without that mutation. So deleterious it's the wrong word, frankly.
Yes, and we had the same discussion because you co-wrote, Rob, and we had the same discussion before publishing the paper. No, you're right. No, you're right, and we agree, and that's why we say, you know, we're just like, you know, and I'm glad we did it and we started, but like I agree with all of you. It's like it's not a clear, neat definitions, and that's why also we decided not to dig any further into this because it's like it seems like still too early to actually have a catalog of deleterious mutation in humans. Okay. Yes, sure. Where the what?
Blood, yeah.
The blood. Oh yeah, the blood. Yeah, sorry. So I was confused. Okay, so we decided to stop here and ask another question. So again, we had this incredible resource. We have admixed individuals. How can we leverage this admixture and to map phenotypes to genome? And so this is an ongoing story, which we haven't completed, but I really like it. So we try to leverage admixture in two ways. Sylvia worked on admixture mapping, and if you're not familiar with admixture mapping, this is a mini lesson. This is a bunch of controls, and this is a bunch of cases. And admixture mapping means and the different colors is the different ancestry of the chromosomes. Let's say this first chromosome, it's of ancestry green, population 1 here, but then this tract come from a different ancestry. And in admixture mapping, what you try to do, you try to see if all the cases share a particular ancestry more often than expected compared to controls. And let's say, imagine like, you just like you do an association between the ancestry and the ancestral distribution on the chromosomes and the disease. And then you end up with like a peak in this position of the chromosome, which translate into, some sort of signal. So this is just a cartoon. So Sylvia did that for obesity. And so we decided to investigate obesity, and we selected 200,000 variants, which are highly differentiated between Africans and Europeans. And we have 115 cases and 900 controls. And then Sylvia ran this analysis, and she finds 4 beautiful signals everywhere, on chromosome 3, 6, 16, and 22 or 21. But mostly interesting, she found a signal in this gene, which is called WWOX. And some of you are familiar with it. And I'll tell you why this gene is interesting. So first of all, this is a cartoon of the gene, which has very long exons. And this is our signal from the obesity. It's surprisingly a small region for like mapping. And that's the power of admixture mapping. That's why we can like restrict. And we have 34 variants here. And this gene, it's important because it's one of the largest, as I said. It contains interesting sites. Contains these sites, which is the chromosomal fragile site in human, like this orange bar that you see here. It's included into a site that in humans tend to like recombine or get broken or something happen. And then also it contains human accelerated region. If you're not familiar with that, and we actually have one human accelerated region within the where we have the signal. If you're not familiar with that, human accelerated region are region of the genome which are incredibly conserved, except for humans. And so people believe that they might have something to do with the fact that we humans are not apes. So that's an interesting place. And we haven't, what we're doing now is to try to replicate the data in all of us. Good luck with that. And try also to evaluate the effect sites. And I think, Sylvia, you did evaluate the effect sites, but we're not showing it here today. But meanwhile, what happened is that Franco was working on something else. So keep in mind the story. Sylvia worked on obesity in European African, and she got a signal in WWXO. And then Franco is working instead on detecting natural selection, leveraging admixture to detect natural selection. And the principle is the same. So what you see here is a region of the genome, and we have a test position, a test SNP, which is this dashed line. And the whole game is to see if what you see here in purple and green are ancestry segments, haplotypes, and they are sometimes, they're very long. In the purple ancestry, they tend to be very long compared to the green ancestry, and so that means they tend to be conserved. Like recombination doesn't happen very often there. They tend to be conserved, and recombination happens, but then among similar. Well, we all know we're geneticists. We know that. So what happened? The game is to find how fast the length of this ancestry tract decays. And if there's a difference between the two ancestry, you see this bubble here. If not, the curves overlap. And so you do these for windows in the genome, and you do for thousands, millions of windows, and that's what you get. And it's very similar to a Manhattan Plat, but what happened is that whatever peak goes on the top is an excess of European ancestry, and whatever peak goes on the bottom is an excess of like a Native American ancestry. Franco was working on, while Sylvia was working on European African, Franco was working on European Native American admixture. And what it detects is a signal in WWOX, which indicates that there's a selective pressure to keep Native American ancestry a WWOX. And you say, oh, that's just a Manhattan plot. No, this is like a magnification of the signal, but Franco actually ran extensive simulations, and we determined, there's no time to go into details, but we determined that the admixture time took place like 12 generations ago, which is like a kind of positive controls, because we know that. But we determined a selection coefficient, which is pretty consistent. And if you're not familiar with the range of value of selection coefficient, this is like in the range of like very strong selective signals. So to go back, we got this Franco get to the conclusion that we have pressure for retaining American ancestry there. And then we tried to put this thing all together. So Sylvia found access, like it's good to have African ancestry, a WWX instead of European ancestry. And Franco says, oh yeah, it's actually pretty good also to have American ancestry. And then we found a paper that says, oh yeah, we studied a different population this time. So because 1 and 2 are both in BAG, but 3 is in a completely different population. And they say, you know what? Also, we analyze, we considered European African admixture in a different population, and we also concluded that it's better to be African there than European. And so we call this project Everything Except Being European and WWX, because we haven't found a title yet. But what we want to do, and this is going to be, as I said, this is unfinished. What we're going to do is to figure out why it is actually good not to be European there. And I hope, we make a nice publication out of it. Okay, so conclusion from this first part is, I'm so proud that, the biorepository is up and running. We're close to 50,000 individuals. We analyzed 13,000 genomes, and we see very complex pattern of ancestry. We see they're different from usual reference, what we usually, we're able to see. And also we found a lot of novel variants which have clinical relevance, and they're not represented in current databases. So we do have a very interesting population. I didn't put any conclusion on the admixed part, but you see that, I hope I convince you also that we have a chunk of our population that can be used in a way to leverage admixture. Okay, question, move on the second part. Please go ahead.
So there is a natural selection of pressure to retain the related genes for African Americans. Have you compared to African? There's like people who can survive.
Yeah. Totally.
Can survive in the United States, but then
Yeah, totally. This is where we're going. This is like the follow-up that we're planning to do. It's like if these are so like selective pressure is interesting because something that might be advantageous today in this environment might not be even more advantageous in another. So the question is to understand if, and usually, so for selection to take place, it's usually most of the time it's what is called selection and standing variation. That means that we do have a repertoire of genetic variants, and that at one point in the demographic history of a species, they become useful. And so, because like, if you think about when a new genetic variant is created, it's just like it's one in one genome. So by the time it expands, it takes forever. And because we humans, we're boring and we reproduce every 40 years, I know it takes forever. So, it's something that we cannot observe in humans. We can observe in microbes and in other species, but not in humans. Most of the natural selection events that we observe in humans are on standing variation. That means like selection just take place. And you can read about this. There's a lot of like nice work about selection going on in Tibetan population, adaptation to high altitude. It's beautiful to see how selection is an action in human, but there's very few cases. And we hope this might be one of that, but that's too ambitious. Let's see. Okay, so we've done all the work that we have done that I've represented so far is based on inferring the ancestry of the individuals. And then we figured out that that's good, but you can do something better. And Franco actually has been working in the past, in his past life before GGI, he was working on identical by descent, and he was very interested in that. And so we were start talking about this. And if you're not familiar with the concept, just follow the orange chromosome here. So this is generation 1, and everyone has his own chromosome. And then people just mate and they admix, and the chromosome get, admixed at recombination. And then what happened that the same orange chromosome is inherited in it recombines here, and then you don't got the whole orange, but just you got pieces. And it's also recombined here. And then this is like a first cousin mating, but just like to make it shorter. But what happened is that you can find the same orange segment in like descendant of very like remote ancestor. And this is what is called identical by descent, because it's actually coming from the same ancestral chromosomes. So IBD, the game changer concept is that identical by descent, it's not only between first cousins, but it's between everyone. Like even you and I, we do share pieces of genome identical by descent just because we're humans, right? And so like families share a lot of identical by descent, and then individuals in the same population, like same geographical population, share some more, some less, compared to like 2 African individuals share more IBD compared to, one African and 1 European individuals. And then third level, and I was practicing this morning with the student, thank you student. Third level, we all share something because we're human. So we just like share something. And so that's exactly this, in this work that I'm presenting after, we focus on this like long range, like long distance identical by descent, which is still incredibly informative. We had concluded before, if you were not sure about it, that assign label to individuals is not easy. So this is IBD in pairs of individuals from BIG. And what you see here is that we piled them up into classes, people that self-reported in the same race, or pairs that self-reported in a different race. And of course, like monozygotic twins, they're self-reported in the same race. And you see all the range of possible parent, possible relationship here. But this is people who self-reported in different race. And it's striking that some people which are related almost to first degree, they self-report in 2 different races. And let me, if you're not convinced of how difficult it is to assign and classify individuals, let me show you this. When we pile them by ancestry, so this is the IBD, and every dot here represents a pair of individuals, and they are piled by their inferred ancestry and colored by self-reported ethnicity. And look at this triangle here. This is a pair of individuals which are related to almost first degree. They both self-report white, but we infer one of them to be African and the other one to be admixed African American. And so it's just like it doesn't work. We can't use labels. It's just confusion. So we avoid using label at all. And what Franco did was to compute identical by descent between all possible pairs of individuals in BIG, which is a massive calculation. And this is the distribution. That means like few individuals share a lot of IBD. These are like the twins or the relatives, and most of the individuals share, very little IBD. But you can use this information to build a network. And the network that you see here is a network where every dot is 1 BIG individuals, and the connection is proportional to the similarity. The closer the dots, the more similar are the individuals. And then this is fine and nice. You can't actually say much about it. You clearly see some clusters. But then what Franco did was to apply an algorithm that takes the network and makes community, like you aggregate the dots into community, and that's like like through several iterations. And this is the results. So from the IBD network, we were able to identify 4 large community, and then he ran a second round of community detection, and it got within community, and we identified 17 sub-communities. And you see here in color, so this is community 2, and this is community 1, 4, and 3. And community 2 is, and what you see here is also like the sites of the sub-community. So community 1 is the larger in size, but community 2 is the one with the largest number of sub-communities. Can you guess whose community 2 is community 1? Would you like to guess? Okay, I can tell you. So we, of course, we compared, oh, Franco, this is C3, C2. Of course, we compared the inferred ancestry with the community definition. And it turned out that community 2 is mostly European, and community 1, sorry. And community 2 is composed, this is interesting. Community 2 is composed from mostly of African individuals that we infer to be African, or they infer to be admixed European Africans. And what you see here for community 1, 2, and 3, and we can't show it for community 4 because it's too few data, is the connectivity and betweenness. Those are 2 measures that tells which individuals are like hubs, are driving the community aggregation. And in community 1, this is made by European individuals, and in community 2, it's equally made by either individuals that we classify as African or admixed European African, and community 3, individuals of Native American. If you're curious about how these relate with reference population, this is a joint set of 1,000 genome and AGDP. It's a very well-known panel for humans. And this is the IBD of our individuals with the 1,000 genome and AGDP now. So it's like how much individuals in community 1 or community 2 share with individuals of African origin in AGDP and 1,000 genome. A lot. Like, we clearly see that community 2 correlate with individuals of African origin, community 1 with individuals of European origin, and so forth. And we also see interesting things like this contribution of a Surui population, which is a population of Native American origin, to one of the sub-community in the African compound, and that's because that means that sub-community reveal fine demographic history. It might be due to just one random event, just people that happen to live, close and admix. So that's really powerful. If you wonder how community relate with self-reported race and ethnicity, exactly as expected. Community 1 self-report white, community 2 self-report Black or African American, and so forth. Community 3 and 4, they're a little bit of mix and match of all the ancestry. And about ethnicity, everyone is pretty clear. C1, they say they're not Hispanic. And community 2 too, and then there's the Latinas, which we always make jokes because Franco is from Argentina, and we make a lot of jokes about that. And so they kind of feel they belong. Some classify as Latinas, some don't classify as Latinas, and so forth. Just to say, so what's the purpose of this? So the sub-community that we identify, they actually corresponds to other type of classification that people have been using forever. It's just this is a way to classify them without putting labels. And also, we think it's more fair because for instance, the European African, their genome is very similar to other African individuals, and that's where they are. So it's it's really interesting. Okay. Few slides more, bear with me, because that's the most interesting part. So we found out that people that are related tend to live together, and they tend to live in the same geographical space. And so this is a nice plot that shows how, of course, first and second degree relatives, they actually tend to live together, but also like third and fourth. So it's like people tend to stick to each other. And you can see it clearly here. This is the prevalence of subcommunity C2A, E, and H. And they do live in close spaces. And those spaces are also complementary. Like each subcommunities occupy its own space. And this is even more, okay, I'll go back to that in one slide. So actually we decided to ask, what about this shared environment? You know, they occupy different geographical locations. So are they, you know, different? And so differential exposure to environment. And so we take the social determinants of health. And if you're not familiar with it, these are like some factors that can affect your health, but they're not like medical condition or genetics. It's like your education, exposure to pollution, and so forth. And this is like a matrix that shows the distance or the similarity between community and subcommunity. And what you see here is like two striking things. The first one, there's a lot of similarity within community, that there's a lot of difference between community. That means each community is really alike, you know, they have the same exposure to what is called social determinants of health, and they're different from the other community. And the other thing is that the distance between subcommunities within C2 is actually, you know, there's some variety here. And we put it next to this matrix, the matrix of identical by descent. And what is outstanding here is community 1, where people is really, really sharing a lot of genome. They're really alike, right? And they make, whereas like the community 2 has less share identical by descent, which, you know, reflects more genetic diversity. So what we're saying here is that people who is similar, they live in the same geographical territory, and they are exposed to the same, you know, pollution, environment, they're exposed to the same environment. And actually, this is really striking. You probably can see the faint green here. This is like, I don't know if you can see the green, but this is the distribution of subcommunity in community 1. The average distance between individuals is 88 kilometers. And if you ever drive to Rob's house, you can see that. And whereas in community 2, the average distance between individuals is 18 kilometers. However, like, people in C1, they live far away from each other, but they really share the same environment. That means, you know, the environment that they share is similar. So does this translate in differential exposure to risk factor? Yes. This is exposure of subcommunities to pollution, to smoking, social vulnerability, and like, you know, and you clearly see the difference here. Like community 2 has greater exposure to pollution and greater incidence of smoking. And another question, does this translate into differential rate of disease? Yes, it does. So this is, we have more example, but this is, the bottom row is disease that are mostly of like genetic origin, and the top is diseases which are not necessarily, but more related to environmental conditions. And you see the difference here, like there's the same prevalence, but here some community have a higher rate of respiratory disease, dermatological disease, of infection compared to other community. If you want to look at this yourself, and here's the, I think like something I'm really proud about, you can go on the web and just click on this app that Franco created. And what you can do is just also evaluate correlation of those things. So for example, this is a snapshot. This is the prevalence of neurological disease, and this is the prevalence of respiratory disease compared to the exposure to pollution. And what you see here in color is the different communities. And for neurological diseases, there's no correlation between being exposed to pollution and developing neurological disease. Whereas for respiratory diseases, there's a high correlation in between like being exposed to pollution and developing respiratory diseases. And guess what? This exposure is not uniform. Some community are more exposed than other. So we believe that this is something that we can really give back to the big community. Like, you know, we're always talking about We got, samples from participants, we got their consent, and we're always thinking about how can we give back to them. This is a way we can give back, because like this instrument that we created can actually inform like public health. It can be taken, like I was talking with the students before, like the City Council of Memphis, they do have data on social determinant of health, but they don't have data on how this relates to disease. And we do provide that. We're saying that, yes, the pollution is differential in the territory of Memphis, and different communities are differentially exposed, and you might know that. But what we're saying is that this also causes differential prevalence of diseases, and this is a fact, and that's something that can be addressed. So you can go on the Shiny apps, and I don't think there's time to go, but you can now. Well, I really want to do that. Let me do that. Because there's another part, but it's boring. There's only one slide. Oh, thank you, Andrea. Okay, well, the app, I think we had it here. So this is open data, and this is like we are releasing aggregated public data of BIG, and this is available for everyone to use. So this is the dashboard of the app that Franco created. And if I go on, where should I go, Franco? Map. And like, you can enjoy and like try to visualize the prevalence of obesity. And I rehearsed this with the student this morning, and smoking. And you see the prevalence overlap, and you can also, whoops, sorry. And you can also see that they correlated. So this is a little bit, I'm sorry, I zoomed into, went to Chicago. Wow. Yes. Yeah, and you can evaluate the correlation, and you can do this for also, you want to say prevalence of obesity, for instance, in community C1D. Thank you, Lisa. And C1D, we also believe it's really amazing. And C1D, and like you can see that it correlates to C1D. And the dots here are actually zip code aggregates of data. So it's like a bunch of, but also let me show off. It's actually Franco's work, and I really kudos to him. He did a great job. What you see here, this is the community, and you can correlate two variables. For instance, I don't know, unemployment and mental disease. Okay, it doesn't. Which was the example that was nice to show, Frank? Oh, exactly. Pollution and respiratory disease. Yes. And you see here, like, that there's a correlation between the two, and that different community, they are differentially exposed to that and differentially like subject to that. So this is really great. But let me finish the seminar. There's one more slide. Yes. So this is publicly available, and we're proud of that. We also tested if this IBD can actually be used in statistical genetics. Like, can we really use it? Yes. We didn't test this extensively. We just want to provide a proof of principle. And this is like a regression model for chronic kidney disease, where we use age and sex, and then age, sex, and IBD, and age, sex, and ancestry. And the IBD performs as well as the ancestry for like modeling. But we believe it captures also the environment, and also there's been a debate about using PCA component derived from ancestry. And also, we test the same in two other conditions, asthma and dermatitis, and we just like. So this is 3 different predictors, and IBD seems to perform slightly better. And if you wonder if 0.5 is actually really different from 0.2, yes. Franco was like, I mean, I'm not an expert in statistical genetics, but we did extensive research, and this is like significant. So it actually can be used. But this is something that, we don't have the expertise to do this in my group, but I'm happy if anyone wants to contribute and help with that, that'll be great. So the conclusion from the second part is that genomic segment, which are identical by descent, actually capture population structure. The IBD communities reflect geographical ancestry and also provide fine-scale structure details. And also the sub-communities are, and also they give the main message that sub-communities are differentially exposed to disease and pollution and everything else. And we really believe that this can be relevant for healthcare. So this is just a list of a number of projects, a number of people that is working on BIG data so far. And just to show you that there's a lot of people working on this data, and we just encourage to do that. And this is just to say thank you to, oh wow, there's a logo here that did not appear, but it's East Tennessee University. I'm sorry. So we're collecting, so the first, of course, gratitude goes to donors, and we're collecting in Tree Pine, Lebanon, Regional One Health, and the East Tennessee University, whose logo disappeared. We do this in collaboration with Regeneron, and I just want to say thank you to all this bunch of people here. And thank you to my group, and also we are sequencing some sample with Ultimate Genomics, and also a little bit of advertising. We're having upcoming 9th edition of the population genetic course, which is great, in June in Italy. And then we will have, I'm proud to say it, with Andrea and Eric and Piotr and Nadia Pisanti, we are co-organizing the first edition of the EMBO practical course in population in pangenomics. In the future, I want to fuse them and do population pangenomics in 3 years. And also we have like MEMPANG upcoming on May 2026. And just to say that this is, speaking of communities, this is to build a community, and just like thank you to all to the large community that we build here in Memphis. Okay, done. Questions? Hey, Eric.
These pretty stuff we haven't talked about in a long time.
I don't know.
But
Sorry, I'm just distracted by checking. Go ahead.
so okay, so you made definitions about admixed and non-admixed, and I was curious if you actually have a few other groups.
If you what?
If you were to change that by an order of magnitude, you would get like 3 other groups in your set.
Yes.
Which would be like European, African, like old admixture, European, American, old right? And probably the same in like what you were pure African.
Yes,
have a similar kind of
totally. That's why we stopped doing that, because it's like it goes nowhere. So like classifying, so the first part of the seminar where I was talking about like inferred ancestry, everything depends on the definition of pure population, and which are the pure population. It's just like hilarious. It's just nonsense. So it's not. And
well, I wasn't saying it was nonsense actually.
No,
those specifically would be groups where an admixture event might have happened longer in the past, which could be interesting.
Yes.
That's one, yeah.
Yes, but what we're trying to do, what we'd love to do is to, so the plot I show about betweenness and connectivity was to start investigating how can we detect admixture from the IBD. And so Franco was trying to question where the admixed individuals falls within the network, within the community, because we believe like, so yes, the more reference population you put, the greater, definition, and also the definition of admixed. We said individuals with at least 20%, 15% in two different, in one answer. I mean, they have 2 ancestors in one, at least 15%. But this is, we got a lot of they were right. We got a lot of critics about how do you define this threshold, and we reply with like, there's a huge debate about that. We don't like it. It's just like, what people do, and it's a balance between come si dice? Reliability, the marker, the power of the marker to detect the tract, like in a consistent way. And so we did a balance between how many markers we had and what's the length of the tract that is reliable. I say that, reliable. And it's just like, yes, it's a question without answer, Eric, because you can keep grouping. It's like fractals. You can keep grouping forever.
It's not fractal. There are Europeans that in your set that are 100% of European ancestry as far as all your reference populations are, and then there are ones that aren't, and the same. So that's there is a threshold of some kind.
My
Are those groups actually different? Do those relate to the subcommunities in those groups that you find
Say it
later?
again?
Do those relate to the subcommunities in the groups later? that
That's
you
what
find?
we had. That's what we started investigating because it's very interesting. And the reason why I started answering with the this thing about the reference population is because the classification depends on the reference population, and that's the major bias. Like it should not be like that. So we should just like. That's true. But Franco is working on applying this approach to the HPRC, and so we're contributing to that.
Oh, sorry. Really great talk. I had a question about the big
Yes.
repository. Have all the cases received a genetic diagnosis, or are there still some that were unsolved?
No, they are, so let me clarify that. We don't, no, that's very good question. So we do see we don't see the patients. What happened is that we have a consent team that every day goes in the hospital and they they talk to people and they say, would you like to share your data and your leftover blood with us? And they say yes. And then we get the data directly from the stream, directly from the hospital to our servers, and we work on that. We do not do diagnosis. And if they've been diagnosed with something, it's by the doctor, and they actually, and this is going to show up in their electronic health records. But that's a good question. We don't do that. We have in mind to do something about like undiagnosed cases, because now those are pretty rare, and because we have large numbers, they're showing up in our database. So that's something that we can definitely think about.
Hi. So you showed a correlation between C2 and community 2 and the respiratory disease, right? Because of the environmental factor.
Yeah.
But is it possible that there are some variants because of which this C2 group is more prone to this kind of disease? Have you studied that?
Sorry, I don't think I got the question. Could you say it again?
So the C2 is correlated with the respiratory disease because
Yes.
you said the environmental factors because the environment is a different
Oh
source.
yeah, got you.
So are there any variants
Yes.
that make them more prone to
Totally,
this?
totally. This is a very complex interaction of like, this could be, this is what we observe. Correlation doesn't mean causality, right? It's just like what we observe from the data. And then definitely there's an environmental component, but it could also be something else. C4 is not plotted probably. Or is it plotted? Or it's under something, yeah. Yeah, C4 is too few. And we have problems showing results about C4 because there's privacy concern about if it's too small, like you can't actually show up. So it's like sometimes disappear. We need more Latinos.
I love it. This was an awesome talk. When Franco did the recombination rate variation, does he correct for the known female bias in maternal bias and the female bias in recombination rates? So maybe,
Oh, in GFAT population sites?
yes.
How did you spot that? Rob, amazing. Well, this is a concern. We haven't. Wow. No, I'm impressed, Rob. Wow. This is a concern. We haven't, and we are in touch with the. No, we haven't.
There he is.
There he
Not
is.
in Argentina.
No, Rob, I'm impressed. You're very fine geneticists. I'm saying is that you really spot the thing. No, we haven't, and we are about to ask to Browning, because I think using the Browning methods how to do that, and if we have to take into account. But yes, that's
It's such important work. There are two themes that I would have liked to have added, and that's just kind of a coda on what you were talking about, health diversity and effectivity. And that is, if you have a population that's well balanced in terms of admixture and ancestry, but one lives here, and maybe you don't have the population size. So one lives in South Memphis, others live out with me in Southwind. What a difference in exposure.
right.
What does it actually?
What
look
is it
like
actually?
medically?
Yeah.
Because it's kind of tempted when you have the correlations to say, what does the residual mean in terms of impact? But I don't quite know how to think about it.
Yeah, no, but and I'll tell you more. That's what we're thinking about like extending this to the CAG, because like we observe, and even in the paper, we always say, this is what we see in Memphis, right? But how this relate to, oh, sorry, this was about the exposure, but in general, about the exposure, how this relate to other population, not a territory, and like, yes.
And it's so important because I think a lot of people that think if you're doing genetics, you're ignoring environment. But no, you can be doing exactly the opposite.
No, that's
Focus.
that's incredible important, and that's what we think.
You had one slide where you put the C2 group, C2E was down in South Memphis and another C2 group was way up in North Memphis. And that kind of shocked me that.
Yes.
So let's C2A and C2E, how do those fall into the Yoruban, Jung?
Oh, I'll tell you.
Isan.
It's
Baika?
CT2. Hey, wait a second. Is which one is the one with the Surui? E and what was the was it E and sorry, sorry, let me go back to C2E and H.
It's an amazing difference.
Yes, and H. Oh, they kind of like, similar IBD with other African. So it's just like they live in different place. They just like live in different place. They're probably the same origin.
Oh, they are from, so, but then I don't understand the map because what is the map showing us?
Yeah.
I mean, I thought it was based on ancestry.
No. The map,
C2E is North Memphis.
yes,
C2A.
H.
No,
A.
C2A.
It's
is
South Memphis.
And there's no overlap between.
No. It's because so if you take all the community two and you take all the community two and you run another iteration of like trying to subdivide them, you subdivide into subcommunities, that's what we have. And then you ask, where are those individuals? Where do they live? Right? And it turns out that each subcommunity lives in different places in community two. Oh, it's not genetic. Oh. IBD, relatedness. Oh, no, in between them.
So in other words, over the last 100 years, that community has been, okay, I see,
Yes.
they're making their own local IBD.
They're making a lot of it. Yes, it's in between that. And actually in the first, well, so in the first version of the paper, we have done the IBD, including also reference population, but we decided to take them off completely because they generate confusion. And I wonder, Franco, if we should take off the where's that the IBD? Where's that this one from the paper, because it might generate confusion. So the IBD all the work is about like shared IBD between BIG community. And then just for fun, we evaluate IBD with reference population just to see, if they're similar to them or not. Okay.
And genomics coming.
It's already there. So Akram has been working on calling the genetic variant with Pangini, which is like pangenome-derived methods. And Lara is learning to compare how these relate with like traditional VCF. And so that's, it's coming. We already did for the first 1,000, and just like we are